CSEC Biology Paper 2 Broad Topic Speedrun. Let's go! Let's start with plant growth. So growth is defined as the permanent, typically irreversible increase in size of an organism by the increase in dry mass and the number of cells. If you're talking specifically about plants, plant growth can be determined by the following: the height of the plant, which is the growth of the meristems, the length of the roots, and also the growth of lateral roots, as the roots that come out the side of the main root, the number of leaves, the thickness or the diameter of the bark, and the presence of flowers and fruits. All of these can be used to determine plant growth. Phototropism. Plants undergo a process called phototropism, which enables them to grow towards sunlight. This occurs on the shoot tips. This maximizes exposure to sunlight and thus increases the rate of photosynthesis. Hormones called auxins enable rapid cellular growth on the shaded regions of the shoot tips. This enables the plant to bend towards the sunlight. This is called anastic growth, which is reversible because they can actually change direction, and it occurs as a response to a stimulus. Here we can get a closer look of phototropism. Here we have a box with a small window here to allow sunlight to come in. The plant, the stems, are going to bend towards the direction of the window. So taking a closer look here, this is what's happening. The growth of the cells will occur fast on the shaded region, that is the part that is not exposed to the sunlight. Auxins are going to accumulate, are going to gather in a great concentration on the shaded region, as is seen here. This is going to allow the cells to divide much more rapidly, and the left side is going to be much more heavier with cells or stem tissue than on the right side. This is going to allow the plant to tilt towards the sunlight. So auxins stimulate growth on the shaded regions of the shoot tips. This facilitates rapid cellular growth via mitosis. The shaded region becomes heavier with cells and tilts the tip towards the sunlight stimulus, which will be towards the window. This is called a growth movement, or sometimes a partial movement. And for animals, on the other hand, animals exhibit whole movements or locomotion. Basically what this means is that a plant cannot actually get up and go towards the sunlight. It must only bend part of its body. But animals, on the other hand, can get up and change their location. This is called locomotion. And both of these are also reversible. Let's take a look at this series of setups here. Now the illuminated side, which is the side where the light is hit, is on the left. The shaded side is on the right. The control, which really has no alteration, is going to bend to the left towards the sunlight. When the tip is removed, however, because auxins really only stimulate growth on the meristematic tissue on the shoot tips, when the tip is removed, there is no phototropic movement. Same thing as if the tip is covered by opaque cap, because there is no stimulation by sunlight here. But if it's by a transparent cap, the sunlight can penetrate the transparent cap and still interact with the tip. If the base is covered, again, no difference because the tip is still exposed. If the tip is separated by a gelatin block, the hormone auxin can still diffuse through the gelatin block and still interact with the tip. Now it's separated by mica, which is a type of silicate, is impermeable, unlike the gelatin block. The auxin cannot diffuse through this. This is why this stem will not bend, it will keep going straight up. Now let's look at gravitropism or geotropism. So plants also respond to gravity. This facilitates maximum exposure to water by the roots and sunlight by the stems and the leaves. So this is called gravitropism or geotropism. So roots are going to have a positive response growing in the direction of gravity. So roots, even if you plant the seed upside down, the roots are still going to grow down. Shoots, on the other hand, have a negative response to gravity. So they will grow in the direction opposite to gravity. So in the case here, shoots will always grow upwards and roots would always grow downwards. In terms of animal growth occurs in almost all the tissues and regions of an animal, whereas in plants it mainly occurs on meristematic regions. This will be, for example, the tips of roots and shoots. As young animals mature, all parts of their body grow until they reach a genetically determined size for each species. The amount of tissue or the amount of cells in the animal will plateau at a certain age. Plant growth, on the other hand, continues throughout the lifespan of the plant. It never stops growing. Germination. Germination is the development of a plant from a seed after a period of dormancy. So seeds are formed from fertilized ovules. They are typically dispersed, example by wind or animals, far from the parent plants to reduce competition. The seed contains an embryonic plant and a nutrient storage called an endosperm. So if you was to relate it to a human, the embryonic plant would be like, a little baby inside or fetus, and the endosperm would be like the placenta. The endosperm would contain carbohydrates and oils as energy reserves and proteins for growth. The tester or the seed coat protects the embryo from any mechanical damage or any injury. Eventually, the seed coat is going to absorb water and is going to rupture or burst to allow germination to take place. Within the tester, the seed coat, you have a rudimentary root or radicle and a rudimentary shoot or plumule that will emerge from a small little thin section here called the micropyle. Adjacent to the micropyle is the hilum. The hilum is the scar of the seed, which was the point of attachment to the seed vessel. So an embryonic plant is different from an embryonic human because, well, one, the embryonic human is still attached to the parent via the umbilical cord, but the embryonic plant is in a seed that is separate from the parent. 2. The embryonic human obtains nutrition from a placenta, but the plant or the embryonic plant from an endosperm. And 3. The embryonic plant can sustain itself right after germination, but the embryonic human requires nutrients from its mother after it is born. Basically, when it is a baby, it still needs breast milk and still needs care from the mother. Cotyledons are small rudimentary leaves. This is what you're seeing here. And these develop because they assist with photosynthesis before the true leaves develop. The cotyledons are then shed because they don't need them anymore. You could think of cotyledons as training wheels for the plant. Basically, they facilitate photosynthesis in the very, very early stages so that the plant can make some glucose and starches in its first few days. Now, the germination itself is triggered by 3 factors: warmth or reasonable temperature, the presence of moisture, which is absorbed by the seed in order for it to rupture, and the presence of oxygen for respiration. Sunlight is not needed for germination because remember, the seed would usually be below the soil, away from sunlight. On this graph, we can see dry mass on the y-axis and days on the x-axis. So for the first 6 days on this graph, the dry mass within the seed decreases. You can see this drop right here. This is because any organic nutrients from the endosperm within the seed are being absorbed by the embryonic plant and are being used for growth and development for the radicle, the plumule, and the cotyledons. After this point here, the dry mass is going to start to increase due to cellular growth and cell division within the plant structures. This is just a key reminder for graphs in general. 1. Include a descriptive title at the top similar to the title of the table of the data. So graph showing something, but it cannot just be y-axis versus x-axis. You have to be able to describe what the graph is about. The second point here is to ensure the scale is continuous. So you can see on this graph here is going 0, 2, 4, 6. It's not going like 0, 2, 3, 4. It has to be continuous. So if you're going in 2s, you're going 2s. If you're going in 5s, you're going 5s. If you're going 10s, you're going 10s. And your points must fit at least half of the page on each axis. 3. Make sure and label each axis with the quantity and the unit. Usually you'll find these in the table of data. Remember, the responding variable will be on the y-axis. So for example, stem height would be on the y-axis because that is responding to something. The manipulated variable, for example, time, is going to be on the x-axis. And lastly, if you have multiple sets of data, 2 or more sets, you're going to have to put a key or legend in place. Whether it's a line graph or a bar graph. If it's a line graph, you can do 2 different types of points. You could put X's and then you could put dots with circles. If it is a bar graph, you can leave one unshaded and the other one you can put little stripes on it. Let's look at nutrition. We have autotrophic nutrition and heterotrophic nutrition. Autotrophic nutrition occurs when organisms produce food from inorganic raw materials. So for example, when plants produce glucose through photosynthesis from carbon dioxide and water. Heterotrophic nutrition occurs when organisms depend on consuming other organisms for their food requirements. So for example, a rabbit eating carrots, or this cow eating this plant, or lions predating a gazelle. So autotrophs, auto means self. So autotrophs means self-feeding. Heterotrophs means feeding from another. So when heterotrophs feed on autotrophs, mainly green plants, they receive organic nutrients from their cells. For example, starch and nitrogenous compounds that would have been inaccessible otherwise. So this is because nitrogen and carbon compounds are fixed into the plants via the nitrogen and carbon cycles, and then they are transferred to animals when they feed on the plant parts. Photosynthesis is a process where the producers take in inorganic molecules and using energy from sunlight produce organic substances such as glucose. This occurs in chloroplasts, which contain the pigment chlorophyll. This would be found in leaves. Photosynthesis has a light-dependent and light-dependent stage. Basically, the light-dependent stage requires sunlight, the light-independent stage doesn't require sunlight. Carbon dioxide and water are the main reactants, and glucose is the main product, and oxygen is diffused out of the leaves, and that is a byproduct. So during the light-dependent stage, light energy is used to break the bonds of water. So water is H2O, right? So it splits into hydrogen and oxygen. But using light to split the water, this is called photolysis. Photo means the use of light, lysis means to split. Oxygen would then diffuse out of the stomata, and the hydrogen molecules that are left behind would combine with carbon dioxide molecules, and this would eventually form glucose. Now remember, photosynthesis has a word formula and a chemical formula. So the word formula is carbon dioxide plus water, these are the reactants, would form glucose, this is the product, plus oxygen, this is the byproduct. The chemical formula is going to be this: 6CO2 plus 6H2O would give you C6H12O6, this is glucose, plus 6O2. Remember, glucose is a carbohydrate which contains carbon, hydrogen, and oxygen, and the amount of hydrogen is always doubled. So in the light-dependent stage, light energy is captured by chlorophyll and converted to energy in the form of ATP. So the light splits the water molecule into hydrogen, which would then go on to form glucose, and oxygen, which is diffused out of the stomata. In the light-independent stage, the hydrogen that is produced in photolysis is used to reduce carbon dioxide to form glucose or sugars. An enzyme called Rubisco catalyzes this process. The ATP required from this process comes from the light-dependent stage. Here we have a drawing of a die-cut leaf, and you should know how to draw this and label it and annotate. The annotation should be in relation to how it helps to maximize photosynthesis. So the leaves with a large surface area, or basically a broad leaf blade, helps to maximize exposure to sunlight because the more chlorophyll is exposed to the sunlight. Then we have the petiole or the stalk. This would help to orient the leaf towards sunlight, kind of like phototropism. We have the leaves have a dense network of veins. These veins contain vascular bundles and xylems, and they allow sufficient water to reach the chloroplast. The underside of the leaf have stomata on the lower epidermis to maximize gaseous exchange. Remember, carbon dioxide is taken in and oxygen is let out during the process of photosynthesis. Here we have a cross section, a transverse section of a die-cut leaf. So if you used to make a cut through a leaf and put it through a microscope, this is the layers of cells that you're going to see. So firstly, we have the upper epidermis. So this is a transparent layer of cells. And the transparency helps because it basically facilitates propagation of sunlight into this palisade mesophyll layer, which contains a lot of chloroplasts. The next thing that it has is a waxy cuticle. And this is a watertight layer which will limit water loss to the top of the leaf. Next, we have the lower epidermis, and the lower epidermis is a thin layer of cells that includes guard cells with stomata, and these allow diffusion and transpiration to occur. In the middle here, we have the spongy mesophyll cells, and these are loosely irregularly packed layer of cells with air spaces. These air spaces facilitate gaseous exchange between the palisade mesophyll and the stomata here. Basically, carbon dioxide is going to diffuse in through the stomata, pass through the spongy mesophyll layer, and then go to the palisade mesophyll cells. So the palisade mesophyll layer is where the party is happening. This is where you're going to get the majority of the photosynthesis occurring. And this is because there's a large number of chloroplasts packed here for light absorption. Sunlight is going to penetrate the transparent upper epidermis and come to the palisade mesophyll. So these cells contain very large vacuoles to keep chloroplasts on the outer edges of the cells to maximize light exposure. They're very adjacent to vascular bundles, and these would contain xylem that would enable water to flow from the veins to the palisade mesophyll. And these cells also have very thin cell walls that facilitate rapid diffusion of gases, in the case here intake of carbon dioxide and letting out of oxygen. For any reaction, there's these things called limiting factors. So let's take a look at an example of photosynthesis. So photosynthesis requires the following to occur: energy from sunlight, carbon dioxide and water, a reasonable temperature, and chlorophyll. If any one of these factors was not here, photosynthesis would not occur. If these factors were here but they were reduced, that would limit the rate of photosynthesis. So a limiting factor is one that, when increased or decreased, influences the rate of a reaction. So for example, if I was to, this graph here shows rate of photosynthesis and light intensity. If I was to reduce the light intensity at a point, the rate of photosynthesis would decrease. If I was to increase the light intensity, it would increase. Now this would only occur up to a certain point, and this is called the saturation point. So for example, light actually stimulates electrons to move throughout the leaf that would engage the light-dependent reaction. There's really only so many electrons that can be stimulated and so much chlorophyll that is in the leaf. So there's a saturation point for light intensity. Basically, if I keep adding more and more light beyond this point, the rate of photosynthesis is not going to increase because almost all of the electrons in the leaf have been stimulated already. Almost all of the chlorophyll has been engaged. If the leaf had more chlorophyll, that is when the rate of photosynthesis would then increase. So in terms of carbon dioxide, to test the necessity of carbon dioxide, you can place the plant or some of its leaves in potassium hydroxide. What potassium hydroxide does is that it absorbs and removes the CO2 from the sample. And since plants require CO2 to form glucose, an iodine test will come up negative for the leaves. Or plant exposed to potassium hydroxide, remember iodine test for starch. So these leaves here would have a negative test for starch because they would not have been able to form glucose from photosynthesis. So in terms of light, when undergoing photosynthesis, the underwater plant here, the aquatic plant Elodia, releases oxygen as a byproduct. And this occurs with all plants, of course, but with an underwater plant, you can see it as bubbles. So you'll see it as rising bubbles gathering at the top of an inverted test tube. So what you're going to get here is a little oxygen column that's going to keep building. So as light intensity increases, the rate of the evolution of these oxygen bubbles will also increase, which means you'll be seeing the more of them coming out per second or per minute, because photosynthesis is occurring at a faster rate. However, past a certain light intensity, where I said the saturation point, the rate of the evolution of oxygen will be the same throughout. So at a certain point, it's going to plateau. Testing for presence of starch in leaves. Well, excess glucose is converted to starch, and this is stored in leaves, roots, shoots, stems, and fruits. The presence of starch indicates that photosynthesis has occurred because the main product of photosynthesis is glucose, and said it's eventually converted to starch for storage. So first you place the leaf in a water bath, and this is going to break down the cell walls. That's what the heat is going to do. And if you have a variegated leaf, remember only some parts of the leaf are green that contain chlorophyll, other parts might appear white. Next, we can boil the leaf in ethanol to remove chlorophyll, and this would make color changes easier to see. So the leaf is going to appear kind of paler green or whitish color. Next, you're going to, using a dropper, you're going to add a few drops of iodine solution. This is going to test for the presence of starch. If starch is present in certain regions of the leaf or in the leaf itself, it's going to turn into a blue-black color. So regions where you have chlorophyll and where photosynthesis would have occurred, those parts would have a blue-black color. The parts that don't exhibit photosynthesis or didn't undergo would remain a kind of yellow-orange color, which is the color of iodine solution. Let's look at cells. Well, the cell is the basic unit of life. cells have organelles such as the mitochondria and the nucleus, which would perform specific roles in the cell. Now, cells can be specialized and they can differentiate their structures to perform specific roles. For example, sperm cells have flagella, which is like tails, that allow them to swim towards eggs so it can fertilize the egg. So the structure usually assists with the function. Eukaryotic cells are the cells that we're more accustomed to seeing. These are found in protists such as amoeba and multicellular organisms such as plants, animals, and fungi. They have a true nucleus and they have double membrane organelles such as chloroplasts and mitochondria. Prokaryotic cells, on the other hand, are found in unicellular prokaryotes. Example, this would be bacteria and archaea. And they do not have a true nucleus. Instead, what they have are loose DNA or DNA plasmids like circular DNA. And they don't have any double membrane organelles, so they don't have any mitochondria or chloroplasts. Here we have an animal cell. We can say that the animal cell has a blob-like irregular shape. This has a nucleus, and this contains genetic material, DNA and RNA, and regulates cell activity. There's mitochondria, which is the site of respiration or ATP production. The jelly-like cytoplasm, that is the site of chemical reactions. There's a cell membrane or plasma membrane that facilitates entry and exit of some molecules. This is partially permeable. Examples of molecules that can enter would be oxygen, glucose, water, but larger substances such as, let me say, another cell like red blood cells cannot enter the cell. Animal cells contain small temporary vacuoles, and these help to store water and any dissolved minerals. There's the endoplasmic reticulum or the ER, and these help package proteins, and these are surrounded by ribosomes which help to synthesize proteins. Also within the animal cell, there would be sugar storages called glycogen granules. These would be deposited in the cytoplasm. Plant cells have a more regular, elongated shape. They contain much of the organelles from the animal cell, which includes the nucleus, mitochondria, the cytoplasm, the cell membrane, ribosomes, and the endoplasmic reticulum, and vacuoles. The difference now is that the vacuoles are much larger and they are considered permanent. And remember, this is because plants cannot actually exhibit locomotion. If they need water, they can't just get up and go and drink water. So a large amount of their water must be stored in the cells. Other structures or organelles that are not in animal cells but only found in plant cells would be the chloroplast, which is the site of photosynthesis. Now, how you can tell the chloroplast from the mitochondria apart is chloroplasts actually tend to be bigger, but they tend to have these striped structures inside, but the mitochondria have folds, inner folds. There's a cellulose cell wall, and this would support the structure of the cell and would maintain internal water pressure in the cell. So when water comes in, it prevents the cell from bursting or undergoing lysis. Starch grains are found throughout the plant cell, and these are energy reserves. Alright, so let's have a rundown of the organelles in the animal versus the plant cell. Well, the nucleus is found in both, the mitochondria is found in both, so is the cytoplasm and the cell membrane, but the chloroplast is only found in the plant cell. The cellulose cell wall is only found in a plant cell. The ER and the ribosomes are found in both. The energy storage in the animal cell is glycogen granules, when the plant cell it is starch grains. And the vacuole in the animal cell is small and temporary, and in the plant cell it is large and permanent. Cell specialization. So specialized or differentiated cells have specific tasks in multicellular organisms, and thus they have features and adaptations in order to assist in performing that task. So below we have a few examples. So first let's look at red blood cells. Red blood cells transport oxygen and carbon dioxide. In order to facilitate this, they have a disc shaped, kind of like a donut without a hole, and this is called a biconcave shape. Basically, the inner part is thinner than the outer parts, and these allow rapid diffusion of oxygen. They also have hemoglobin, which is a protein that binds oxygen, and they don't have any organelles, and this facilitates the maximum carrying capacity of oxygen. Sperm cell, the function is to fertilize an egg. It has a tail or flagellum that allows it to swim, allows it to be motile, and it has chemicals in its head called an acrosome that help it to penetrate the egg. This is a neuron, so the neuron transports electrical messages or impulses through the body. It has dendrites or branches that connect to other neurons, and it has chemicals that help regulate transmission. These are called neurotransmitters. Here we have an epithelial cell, and these line the surfaces of organs. They're usually very thin to allow diffusion of materials, and they may be ciliated or may have hairs to increase their surface area. Osmosis is defined as the movement of water molecules across a partially permeable membrane from regions of higher to lower water potential. So water potential is an indicator of the concentration of free water molecules. The more free water molecules, the higher the water potential. Concentrated sugary or salty solutions would have lower water potentials than diluted solutions. Of course, pure water would have the highest water potential. If we look at the diagram on your right, if we look at the left side here, we can see that there's a high water potential here because there's many free water molecules. On the right side of the diagram, we can see that there's some sugar molecules or some solute. So there's a lower water potential on this side because this has a lower concentration of free water molecules. Between here, we have a partially permeable membrane. This is going to allow water to flow from where there's a high water potential to a lower water potential. This is why the level is going to drop on the left and it's going to rise on the right. In this experiment here, we have some viscine tubing which is partially permeable, and this contains a sugar solution, let's say like a glucose solution. There's a capillary tube attached to the solution here, and this is placed in a beaker of pure water. So what's going to happen here? The water is going to move into the tubing from the beaker, and this will cause the tubing to swell. So this is because there's a lower water potential in the tubing because there's more solutes. There's sugar in here where there's no sugar outside, so there's more free water molecules outside and inside, so the water is going to flow into the tubing. So the water level is going to drop in the beaker because the water is going to flow into the tubing, and the level is going to rise in the capillary tube. Sugar molecules cannot pass out of the tubing because they are too large. Remember, it's partially permeable. Let's look at osmosis in animal cells, but first we have to be able to differentiate between three types of solutions: hypotonic, isotonic, and hypertonic. Now, two of these words sound very similar, hypo and hyper. And hyper really means a high amount of something. Hypo means a low amount of something. So hypertonic means that there's a lot of sugar or a lot of solute in the solution. Hypotonic means that there's less sugar or less solute in the solution. So how I used to remember it, you know, when you're small and you eat sugar and you get hyper, I used to remember the hypertonic one is the one that contains a lot of sugar. Of course, if it contains a lot of sugar, that means that there's less free water molecules. In the hypertonic one, the hypotonic one would have a lot of free water molecules because there's no sugar to be dissolved. So in hypotonic solutions where there's a high water potential, basically a low amount of sugar, the solution has a higher water potential than the cell cytosol. So water molecules are going to flow into the cell. This is going to cause the cell to swell and potentially burst. Remember, animal cells don't have a cell wall, so they can undergo lysis and they can burst. In hypertonic solutions, remember, most of the sugar here is on the outside, so there's very little water on the outside. Water is going to flow out of the cell because the solution is going to have a lower water potential. Inside the cell there's going to be a higher water potential. So water is going to flow out of the cell cytosol and into the solution. So water molecules are going to be lost by the cells, and this is going to cause it to shrivel or crenate. So in hypotonic solutions, there's going to be a positive change in mass, and hypertonic, there's going to be a negative change in mass. It's going to decrease in mass because water is going to flow out into the sugary solution surrounding it. So in isotonic solutions, this means that the concentration of sugar and water is the same both in and out of the cell. This one is easy. This means that there will be no net change in the mass. There's no net flow of water molecules in and out of the cell, so it remains the same. The prefix iso means same. So in plant cells, of course, the isotonic solution, you wouldn't see any change here because the net flow of water in and out is going to be 0. In hypotonic solutions, where there's more water and less solute outside of the cell, there's a higher water potential outside. Water molecules are going to flow into the cell from the solution. So the cell is going to become turgid and is going to swell. The vacuole is going to absorb water and then expand with cell sap. In hypertonic solutions, it's going to be the opposite. Water is going to flow out of the cell because the higher water potential is going to be in the cell and the lower one is going to be outside the cell. So water is going to flow out of the cell and into the solution. The cell is going to become plasmolized, and if it was a plant, the plant would become soft and it's going to wilt. The cell membrane is going to retract from the cell wall as cytosol is lost, and the vacuole is going to shrink as water is lost. So remember, the plant cells do not burst or do not undergo lysis because they have cellulose cell walls that help to maintain the internal pressure. You've probably done this experiment in school, but if you're using potato strips, we can see two setups here. We have raw potato strips and two petri dishes. One is filled with pure water and the other is filled with 20% sucrose solution. So in dish A, water molecules are going to flow into the cells from the solution, higher to lower water potential. The potato strip will become turgid and is going to increase in length. So there's going to be a positive increase in length and mass. In dish B, on the other hand, water is going to flow out of the cell because there's going to be a higher water potential in the cells compared to outside. So it's going to flow out into the sucrose solution. So it's going to become flaccid or kind of floppy, and there's going to be a negative decrease in size and length. So in higher molarities, let's say 40% sucrose, the decrease in length will be even greater. Let's look at the carbon cycle, and let's start with atmospheric carbon dioxide here. Now, there's really only two main ways in which carbon dioxide is removed from the atmosphere. One of them is photosynthesis, and this is when carbon dioxide is taken in by leaves to make glucose. The other main one is that when it is absorbed by water, such as big bodies of water, such as oceans and lakes. So let's start with photosynthesis. So atmospheric carbon dioxide levels would drop as they are absorbed by the leaves for photosynthesis. This would fix carbon compounds into the leaves. So animals are going to consume the leaves, and then other animals are going to eat those animals, and they are going to gain carbon into their bodies. When the animals respire, basically when they breathe out, they're going to exhale carbon dioxide, and that is going to return carbon dioxide to the atmosphere. Now, what could also happen is that when the animals die, they are going to decompose, and decomposers are going to break down the organic dead matter within their cells, and that process of decomposition is also going to release carbon dioxide back into the atmosphere. Now, any carbon compounds that remain in the cell, eventually this is going to undergo fossilization, and this decomposed material is going to become fossil fuels. Basically what we would know as coal or natural gas, and this is going to this is going to take place after many years. Finally, the fossil fuels are going to get pumped back up to the surface, and they are going to be burnt and used by automobiles or by factories. And basically when these fossil fuels are burnt, or when they're combusted, they're going to release CO2 back into the atmosphere. The problem here is this process of combustion is what's going to massively increase the amount of atmospheric carbon dioxide. But not just carbon dioxide, but other greenhouse gases as well, such as methane or such as sulfur dioxide. This could pose a serious threat to the environment. So we have three main processes here that throw the cycle off balance. One is deforestation. So if there's fewer trees, there's less carbon dioxide that's going to get taken up by those trees for photosynthesis. So the atmospheric carbon dioxide is not diffused into the leaves, and these carbon dioxide levels remain high. Combustion raises carbon levels due to the emission of gaseous carbon compounds. Mostly anything that uses electricity uses fossil fuels, and those require burning those fossil fuels. Then we have sewage runoff. So excess carbon dioxide would cause oceans to become acidified, and this would threaten population life. This could also just be due to the absorption of excess carbon dioxide into the oceans. This is where we'll get coral bleaching, for example, occurring. Conservation is the preservation of habitats and natural resources so that they remain available for future generations, as well as to maintain ecosystem stability. Restoration is the act of returning ecosystems to their previous normal states following interferences or disturbances. This could be due to human impact, it could be a natural disaster like an earthquake or hurricane. As I said, basically it means returning the environment to its natural state after some kind of event happened to it. So what are some practices involving conservation and restoration? Well, first we have reforestation, which is replanting trees that have been cut down. This will reverse habitat loss. There's also afforestation, which is just planting trees, not replanting. There's sanctuaries, which ensure that endangered species are kept in protected reserves. There's proper agricultural practices, so using natural fertilizers with less chemicals in them, less synthetic chemicals, can restore carbon and nitrogen compounds to the soil. Crop rotation also increases fertility, and banning harmful methods such as slash and burn will limit destruction of soil nutrients. Lastly, there's the reduction of fossil fuels, carbon taxes, encouraging carpooling and public transport, and using more energy efficient vehicles such as hybrid vehicles and appliances would all help to reduce fossil fuel usage. There's also alternative forms of energy that could be set up, such as wind turbines, solar panels, hydroelectric dams, nuclear energy, geothermal vents. All of these here would reduce combustion of fossil fuels. Blood components. Well, blood is a fluid that contains specialized cells. This means that blood is a tissue. The main components of blood are blood plasma, which is a pale yellow fluid that transports soluble material in the blood. So example would be any absorbed nutrients to be assimilated into cells, hormones, nitrogenous waste. Next, we have red blood cells or erythrocytes, and these transport oxygen from the lungs to the body cells, such as muscle cells. They have a biconcave structure that maximizes diffusion. They have hemoglobin, which help to bind the oxygen, and remember, they don't really have many organelles, so they can maximize their oxygen carrying capacity. Phagocytes engulf bacteria and foreign bodies, for example, any antigens or toxins. So it is a large globular structure with a multi-lobe nucleus. They move around using a pseudopodium, which is like an extension of the plasma membrane. And what they would do is that they would wrap around or evaginate any foreign materials and then digest them and then excrete them. Lymphocytes rapidly divide and release antibodies to eradicate pathogens. Think of lymphocytes as snipers and the antibodies as bullets to get rid of the enemy pathogens. Large round structure is a large round structure with a very round nucleus. This is how you're able to tell them apart. Platelets are very small, irregularly shaped fragments, and together with clotting factors, they help to convert soluble fibrinogen in the blood plasma to insoluble fibrin to produce blood clots. Basically, these would help to limit bleeding. So how oxygen is transported in the body is that the air is breathed in through the nostrils, and then the air moves through the pharynx, the bronchi, the bronchioles, and the alveoli, that's the air sacs in the lungs. Oxygen would diffuse through the thin, moist alveolar membranes, through the capillary walls, and then into the red blood cells. Red blood cells would then transport the oxygen via arteries to body cells such as muscles. So capillaries are only one cell thick, and this is to facilitate rapid diffusion. They often connect organs to the bloodstream, and they also connect arteries to veins. So surrounding these body cells here, these cells are tissues, a liquid that is called tissue fluid, and these would help to transport diffuse materials between the cells and the capillaries. Tissue fluid is in direct contact with the cells, not the blood plasma in the capillaries themselves. Sickle cell anemia is an inherited disease where the red blood cells become sickle-shaped due to abnormal hemoglobin. Remember, this here is the typical biconcave structure which facilitates easy diffusion or rapid diffusion of oxygen, but it also helps to store that oxyhemoglobin after the oxygen has bound to hemoglobin. But the sickle cell, on the other hand, has a lot of issues doing that. So their function is affected as they become less flexible and their oxygen carrying capacity severely decreases. Fatigue or tiredness will ensue because they don't have enough oxygen for respiration. Painful blood clots may form, and this would result in edema or swelling in the legs and the arms, basically extremities of the body. So here's a recap on some genetic terms. A gene is a segment of DNA that codes for a particular protein or trait. An allele is an alternative form of a gene. So for example, you could have alleles for brown eyes, alleles for blue eyes. Dominant alleles express their trait over recessive alleles. The genotype refers to the combination of inherited alleles, and the phenotype refers to the physical expression of the genotype. So the genotype is going to be like these letters here, capital B, or common B. The phenotype is going to be the expression of the trait of these alleles here. So capital B, for example, would be brown eyes. Common B would be blue eyes. That would be the phenotype. That would be the observable or testable characteristic. Homozygous genotypes have the same alleles. Homo means same, zygous means of the zygote, means the same alleles in the zygote. So they have the same alleles, so capital B is called homozygous dominant, common B is called homozygous recessive. Heterozygous genotypes, it means hetero means different, have different alleles. So for example, capital B or common B, so they would have one dominant allele and one recessive allele. Specifically about sickle cell anemia, let's look at a genetic diagram for it. So we have hemoglobin A, and this is the healthy allele, and this is dominant. And we have hemoglobin S, and this is the sickle cell allele, and this is recessive. So the question here is, how can a mother and father who don't have sickle cell anemia produce a child who does have sickle cell anemia? So let's look at the Punnett square here. So the father, he would have, remember, each parent would have two alleles. So the father is going to have a healthy allele and a sickle cell allele. So he's going to be heterozygous for sickle cell anemia. The same is with the mother. So if we look here, this first child, now remember, Punnett square describes the inheritance of traits, but it doesn't mean that the mother and father are going to have 4 children. What this means is that for every 4 children that they have, this will be the possibilities. So for every child that they have, these are the possibilities. So basically, imagine that this mother and father have 40 children. It means 10 of them would have this, 10 would have this, 10 would have this, 10 would have this. So think of it as representing a ratio. So let's look at this first possibility here. So this first child is going to inherit hemoglobin A from the father, hemoglobin A from the mother. And this child here is going to be normal because it's, these are the healthy alleles. This second child here is going to inherit hemoglobin S from the father, hemoglobin A from the mother. Now, because hemoglobin S is recessive, this doesn't mean that the child will come out with sickle cell anemia. This child is said to have sickle cell trait, which means that the child is a carrier, still healthy, but is able to pass on the sickle cell allele to their children. The same is going to be for this child. Is going to inherit hemoglobin A from the father, hemoglobin S from the mother. So this fourth possibility here is going to inherit hemoglobin S from the father, hemoglobin S from the mother. This child is going to be suffering from sickle cell anemia. So both parents must be heterozygous, both must be carriers. And the child with sickle cell anemia will inherit the hemoglobin S allele from each parent. So the phenotypic ratio is going to be this. There's going to be 1 normal, 2 with the sickle cell trait, that means that they are carriers, and 1 that's going to be the sickle cell sufferer. So all in all, the probability of a child getting sickle cell anemia from these parents is going to be 25%. So one other note here, normal individuals, so basically healthy individuals who carry 1 sickle cell allele, basically these 2 here that are heterozygous, they actually procure some resistance against malaria because the Malarian pathogen, Plasmodium falciparum, actually lives within the red blood cells. They take in nutrients from the red blood cells. What the sickle cell trait does is that it makes the red blood cells more porous, and most of the nutrients actually leak out. So the pathogen cannot actually survive within the sickle trait red blood cells. Ovulation and fertilization. Here we have a diagram of the uterus. We have the ovaries here. These are the fallopian tubes. And this would be the endometrium or the uterus lining. So ovulation is the release of an egg cell from the ovary into the fallopian tubes. Fertilization occurs when a sperm cell nucleus fuses with the egg cell nucleus to form a zygote. This usually happens in the fallopian tubes. Sperm cells will be ejaculated through the vagina, swim up the cervix, up the uterus, through the fallopian tubes, and finally it will fertilize the egg in the fallopian tubes. The zygote is going to undergo mitosis and is going to divide into a ball of cells called a blastocyst before implanting on the endometrium or the uterus lining. This is where it will develop an amniotic sac and a placenta to receive nutrients and oxygen and antibodies and so on from the mother. This is where now it will develop into an embryo and then a fetus before childbirth, where it will become a baby. Now what we're going to focus here on though is mainly the processes of ovulation and implantation, and finally menstruation. So there's 4 key hormones involved in this. The first is FSH, or follicle stimulating hormone. This egg cell that is released, this is encased within a sac. That sac is called a follicle or a Graafian follicle. This encases the egg cell. FSH stimulates the growth of that follicle. LH, or luteinizing hormone, triggers ovulation. So when there's a surge of LH, there's usually a little spike in the increase of LH. This would trigger the rupturing of the follicle and allow the egg to be released, and this would lead to ovulation. Estrogen thickens the endometrium for implantation. Think of the zygote attaching to the endometrium as planting a seed in soil. Estrogen ensures that that soil is there. Estrogen also stimulates the release of LH. Progesterone maintains the thickness of this uterus lining. So once progesterone levels are high, this would remain thickened. When progesterone levels decline, or when they drop, the uterus lining begins to thin and then begins to shed. This would initiate menses or menstruation. There are 3 main phases in the menstrual cycle. There's the follicular phase, the luteal phase, and at the end of the luteal phase there's going to be menses or menstruation. So let's start first with the follicular phase. So first we have the development of the Graafian follicle. That's what you're seeing on top here. Seeing that this is getting bigger and bigger, and this is what contains the egg cell. So the Graafian follicle develops and matures due to the secretion of FSH. We're looking at the green curve here. We could see as there's an increase in FSH, there's also an increase in estrogen, and eventually there's going to be a surge. We're looking at the pink line, there's going to be a surge in LH. Now this surge in LH is going to trigger ovulation. It's going to trigger the rupturing of the follicle and the release of the egg cell, and this occurs in the middle of the cycle on day 14. This is when the egg is released from the ovary, and this would mark the end of the follicular phase. In the luteal phase, after the egg is released, you'll see that LH levels begin to drop. So if you look here, remember the egg has been released from the follicle, but what happens to the follicle now? The follicle becomes a corpus luteum. Corpus luteum just means yellow body. And the corpus luteum secretes progesterone to maintain the endometrium's thickness. So we could see here, as there was a rise in estrogen, that the cells in the endometrium began to proliferate and this began to thicken. Progesterone is going to help to maintain and to help to keep growing this uterus lining. So as you can see here, when progesterone levels drop, so does the thickness of the endometrium. So progesterone levels will stay high for a few days. This is where it's possible for the egg to be implanted and to become an embryo and a fetus. Lastly, now we have menses. So this occurs at the end of the luteal phase. The corpus luteum would have broken down and progesterone levels would start to rapidly decrease. Remember, the corpus luteum was responsible for the release of progesterone to help to maintain and thicken the uterus lining. So when the corpus luteum breaks down, the progesterone levels drop, and this here would begin the breakdown of the uterus lining. So the unfertilized egg and endometrial material are shed through the vagina, and FSH levels, FSH and estrogen levels, are going to rise slightly as a result. So this here is called menses or menstruation. So birth control refers to any method that prevents pregnancy from occurring. It is usually used by families that wish to avoid the financial and emotional strains of raising a child, and also to limit population growth in countries. So this can occur via two methods: contraception and anti-implantation. So contraception is basically preventing the fertilization of the egg. Examples of this would be condoms, the rhythm method, vasectomies, and tubal ligations. Anti-implantation means that the egg is already fertilized, but the fertilized egg cannot be implanted on the endometrium. So examples of this would be IUDs or intrauterine devices, or emergency contraceptives or morning after pills. So let's look at some categories of birth control. First, we have barrier methods. So condoms prevent sperm from entering the vagina, and diaphragms or female condoms prevent sperm from entering the uterus. Now, these protect against STIs or sexually transmitted infections if used correctly, because there's no real exchange of sexual fluids. In this case, there's no exchange of semen and vaginal fluids. Next, we have the natural method or the rhythm method, and basically this is where the girl or the woman would pinpoint the time of ovulation and avoid intercourse at this time. This is highly unreliable because the menstrual cycle is subjected to change based on environment, based on diet, based on physical activity, based on stress. So this is quite unreliable, and typically there's a high failure rate in this. Surgical methods involve vasectomies and tubal ligations. So with vasectomies, there's the ligation, which it basically means the cutting and tying off of the vas deferens. The vas deferens is the sperm duct that takes sperm cells from the testes to the urethra before ejaculation. So in this case here, the male is sterile, which means it could still ejaculate, but just it wouldn't have, the semen wouldn't have any sperm cells in it. Tubal ligation, which means that the fallopian tubes are tied and cut off. They would prevent eggs from being released into the uterus. So the fallopian, basically like you're cutting off a little bridge here. The eggs are still released, but they cannot be implanted onto the uterus, and they cannot be fertilized because these sperm cells will not be able to reach them. These have high success rates, but offer no protection against STIs. The downside of this is that they are more expensive, especially tubal ligation tends to be a kind of invasive surgery, and they are very, very difficult to reverse. They tend to be irreversible. Hormonal methods basically includes birth control pills, and these can work in several ways. So the modes of action can include that they can suppress ovulation so that no egg is released. They can thicken cervical mucus to prevent the entry of the sperm. It can reduce endometrium thickness or stop the uterus lining from getting thicker so that there's no soil to plant the seed, so that no implantation occurs of a fertilized egg. Now, male contraceptive pills are still in the works, but how they can potentially work is that they can reduce testosterone levels and thus decrease sperm production. They can help to make the sperm immobile. Basically, they can withdraw the function of the flagellum. They can thicken the mucus in the vas deferens as the sperm duct to reduce sperm release. Now, one thing you have to remember about birth control pills is that they have to be taken every day, and usually from the start of the cycle. There could be a lot of hormonal side effects that could occur, such as mood swings, weight gain, and fatigue. Morning after pills or emergency contraception, such as Plan B here, there are various types of them, but the mechanism of action usually involves delaying ovulation or causing changes in the endometrium that limit implantation. So this must be taken up to 3 days after fertilization occurs, despite the name morning after. So other birth control pills must be taken every day, as I just said, from the start of the cycle to be effective. Alright, so we have mechanical methods of contraception. So these involve IUDs or intrauterine devices. These are small T-shaped pieces of plastic or copper which stimulate the production of white blood cells to destroy sperm cells. So some of them secrete slightly inflammatory compounds that, engage white blood cells. And some of them produce hormones that thin the endometrium that limit implantation from occurring. STIs or sexually transmitted infections are communicable diseases that occur when a pathogen is transferred via sexual contact. Remember, communicable means it can spread from one person to another. So examples of this would be AIDS, which is caused by HIV, which is a virus, herpes, which is caused by a virus, syphilis and gonorrhea, which are both caused by bacteria, and cervical cancer, which is caused by the human papilloma virus. So one key thing to note here is that any disease that is caused by a virus must rely on the immune system or monoclonal antibodies to eradicate them. Bacteria could rely on antibiotics. Antibiotics will not work on a viral infection. Here are some key symptoms of some of those diseases. I won't put any pictures of the diseases in case the video gets flagged, but you can easily Google the symptoms yourself. So herpes, there's small red blisters on the genitalia or the mouth, basically the lips. There's itching and pain on the genitals. Syphilis, there's body rashes on the back, the soles of the feet and palms. There's fatigue, hair loss, and weight loss. Gonorrhea, there's a mild fever, burning during urination, and leakage of pus from the urethra. And HIV or AIDS results in rapid weight loss due to muscle wastage, hair loss, dark lesions on the skin called Kaposi's sarcoma, and chronic diarrhea. HIV stands for Human Immunodeficiency Virus. AIDS stands for Acquired Immune Deficiency Syndrome. The human immunodeficiency virus is transferred via semen, vaginal fluids, blood transfusions, and breast milk. It could also be transferred during childbirth. So HIV infects lymphocytes, a specific type of lymphocyte called a T lymphocyte. So those infected cells help HIV proliferate, basically multiply, and infect other lymphocytes. So when the T lymphocyte count drops below 200 per millimeter cube of blood, the patient is said to have AIDS. Basically, the immune system has now undergone a severe deficiency, hence the term. So HIV, being a very small virus, is very difficult to detect and produce a vaccine for. Antibiotics are useless against viruses. Remember, they only work against bacterial infections. And the virus has a high rate of mutation. So how can HIV spread be prevented or reduced? Well, there's the use of barrier methods of contraception, such as condoms and diaphragms, limiting the number of sexual partners or being faithful to a single partner, or in more extreme cases, abstinence. There's regular HIV blood screening, but the thing is about blood screening is that it takes a few months for the virus to actually be detected. So you could get HIV in January and it might not show up in a blood screen until March. So it has to be regular blood screening at least with each new partner that you get. Education on safe sex for the young and the underprivileged, and the use of antiretroviral therapy such as the drug AZT that would help to minimize symptoms and spread. So there's another therapy called HAART that is used by pregnant women, and this would limit the spread of the HIV pathogen to their babies. And those are the broad topics for Biology Paper 2. Good luck.